Hello and welcome to Nikolai's genetics lessons, and the first question is: a charged tRNA attached to the specific place on the messenger RNA where the, and you have to choose between these 5 answers. Imagine that this is messenger RNA, and somewhere here we have ribosome. This is large subunit, this is small subunit, and here we have 3 sites, which are A site, P site, and E site. And here we have 5' end of the messenger RNA, 3' end here. So our ribosome would move in this direction. So the very first tRNA would go to the P site, but all the rest tRNAs would go to the A site first, then to the P site, and would exit large ribosomal subunit through the E site. So let me draw a structure of the tRNA. This is clover leaf structure, actual form that we call L form of the tRNA looks like this. When tRNA is charged, it has also amino acid, and on top of this tRNA also has elongation factors that help tRNA to enter a site. For example, if I would draw tRNA in clover form, it would resemble something, would look like something like this. So one loop here, a variable loop here, another loop here, one more loop. So it would look something like this. And here we also would have amino acid attached to the 3' end. So this end would be 3' end of the tRNA, and this end would be 5' end. If we would stretch this tRNA, we would have 3' end here and 5' end here. So both messenger RNA and tRNA would go in different directions, just like in double-stranded DNA. So the names of these pin loop structures are as follows. Close to 5' end we have D loop, so D loop here. It has it's derived its name from the rare base that can be found here. It's called dihydrouridine, and this loop has a name which is T loop, and it also has rare bases, pseudouridine, and variable loop here, depending on the amino acid that this tRNA would be charged with. This variable loop also varies. And on the messenger RNA, of course, we would find codons. For example, it can be A, or C, U, or C, A, C. As you see, all codons, in other words, we call them triplets, go in 3 bases, and to this codon in the A site, in the tRNA loop, which we call anticodon loop, we would find the sequence that would be complementary to this codon sequence. So we call this loop anticodon loop. So the answer would be answer B. So if we have in codon C here, it would base pairs with G, cytosine with guanine, adenine with uracil, and the third position in anticodon can be variable, and we call this wobble position. So this is going to be third position of the messenger RNA, but first position in anticodon, because tRNA is made from 5' end to the 3' end. So if here it is going to be third position in tRNA, this is going to be first position of the anticodon. And next question, the pyrimidines in DNA are, and you have to choose between these answers. If you're confused which answers to choose, I have a good mnemonic technique for you that also would allow you not only to memorize which bases are purines and which are pyrimidines, but much more information that would be valuable for you on your exam. So here is my technique. First of all, we have to write cut, and as you know, this is three bases: cytosine, uracil, and thymine. Uracil and thymine I put together because in messenger RNA, thymine would be substituted with uracil. So if DNA we have thymine, in messenger RNA we cannot find thymine, instead we would find uracil. And uracil and thymine are almost identical bases with a small change. So also most of you know that cytosine would base pair with guanine, and thymine would base pair with adenine. And also you can easily memorize that cytosine and guanine would make triple bond, because once again, this is not scientific method, this is mnemonic technique. Cytosine and guanine look alike. For example, thymine and adenine, these two letters doesn't look alike, so just like relatives in humans have more connections between them, so cytosine and guanine have triple bond, and thymine and adenine, or uracil and adenine, would have double bond. Now, how to memorize which bases are purines and which are pyrimidines. Purines are bases with two rings, and pyrimidines are bases with one ring. And as you see on the left, we have a word cut. That means that this basis has one ring. So this one ring base, this one ring base, and this one ring base. And automatically now we know that guanine and adenine has two rings. Now how to memorize that cytosine, uracil, and thymine are called pyrimidines. It is also very easy. Purines are short word. Pyrimidines is words that has more letters. So now something illogical may sound for you something illogical. Long word stand for the short bases, and short word stand for the long bases. It might seem that it is hard to memorize, but actually something that is illogical we usually memorize much easier than logical things. For example, if you would see today a black or white cat, you probably would forget about it in an hour. But if you would see a green cat or a blue cat with red spots, you probably would memorize such an event for the rest of your life. So sometimes illogical things are easier to memorize. So just memorize that long word pyrimidines stand for the short variants of the bases, and purines stand for the long variants of the bases that has two rings. So let's now choose the correct answer. Answer A cytosine and uracil seems like correct answer, but you need to pay attention that the question is about DNA, and uracil we only can find in RNA. So this is not the correct answer, but the correct answer would be answer E, cytosine and thymine, and this is answer E. And also one more hint for you, which mutation we call transversion and which we call transitional mutations. So if in a strand of the DNA one purine would be replaced with another purine, or guanine would be replaced with adenine, or adenine would be replaced with guanine, we call such mutations transitional mutations. And this is also true for pyrimidines. If one pyrimidine would be replaced with another one, we call such mutation transitional mutation. But if pyrimidine would be replaced with purine and vice versa, we call such mutation transversion mutation. It is also easy to memorize. If one ring base would be replaced with another one ring base, this would be something more normal than if one ring base would be replaced with two ring bases, because in this case two ring base would base pair with also two ring base. And also it is easy to memorize that transition is something like boy transit to be a man. So this is something normal process. And transversion, it's like similar to transvestite, when male, say, would become a female. So this is something abnormal. So this type of mutation we call transitional mutation, and this type of mutation we call transversion mutation. So I hope this video would help you on your exam. This is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.